Hello, we're glad you've joined us for this live webinar, CRISPR-Cas9, moving from founder mice to phenotyping. I am Judy O'Rourke of LabRoots, and I'll be moderating this session. Today's educational web seminar is presented by LabRoots, the leading scientific social networking website and provider of virtual events and webinars advancing scientific collaboration and learning. It's brought to you by the Jackson Laboratory. Founded in 1929, the Jackson Laboratory, JAX, is an independent nonprofit biomedical research institution. Its employees discover precise genomic solutions for disease and empower the global biomedical community in the shared quest to improve human health. The laboratory is a world leader in mammalian genetics and human genomics and generating the development of scientific breakthroughs and improved therapies with ever greater precision and speed. JAX educates scientists and provides critical resources, data, tools, and services to researchers worldwide. It has a mammalian genetics headquarters in Bar Harbor, Maine, a facility in Sacramento, California, and a new genomic medicine facility in Farmington, Connecticut. In addition to its National Cancer Institute designated cancer center, JAX has initiated a cancer research laboratory in Seoul, Korea. To learn more, visit www.jax.org. Let's get started. This webinar has been approved for continuing educational credits. Please click on the CE button at the bottom left corner and follow the process to receive your credits. You can pose questions to the speaker during the presentation while they're fresh in your mind. To do so, simply type them into the Q&A box, which will open when you click the green Q&A button at the lower left of the presentation window. Questions will be answered after the presentation. To enlarge the slide window, click on the screen icon in the lower right-hand corner of the slide window. If you experience technical problems seeing or hearing the presentation, just click on the support button found at the top right of the presentation window, or report your problem by clicking on the green Q&A button at lower left. I now present today's speaker, David Grass, PhD, Senior Director for Genetic Engineering, Transgenic Genotyping, and Reproductive Sciences at the Jackson Laboratory. Dr. Grass has been involved in the generation of genetically modified mice for more than 25 years. He received a PhD in molecular biology from Columbia University in New York and was a postdoctoral fellow at the Sloan Kettering Institute for Cancer Research, where the focus of his research was molecular immunology. Dr. Grass formerly worked at Xenogen Biosciences, a biotech company previously known as DNX, where his groups were responsible for genetically modified model generation, phenotyping, and in vivo compound assessment. His responsibilities at JAX include overseeing the model generation program. Dr. Grass will now begin his presentation.
Thank you. It's really a pleasure to be here today and to be able to talk to you about the model generation platform that we have at JAX. Today I'm going to tell you about that platform, focusing mostly on the work that we do using CRISPR technology. I'll talk to you about project types and timelines, the experience that we have doing a lot of these types of projects. I'll show you a couple of case studies that hopefully will illustrate things that we think about in doing these studies. And I'll also talk about some of the things that we've learned from doing the number of projects that we've done over the last couple of years. So before I get into the CRISPR technology, I want to just start and put it in perspective and talk about the history of genomic engineering in the mouse. And in the 1980s, we really started by taking the approach of the transgenic approach, which allowed us to do gene addition, so to add a gene to the mouse genome. And the technologies that were used to do gene addition were generally pronuclear injection or retroviral infection of embryos, of single-cell embryos. And I guess the most utilized technology was pronuclear injection, and in which you would inject into a single-cell zygote at the pronuclear stage a transgene fragment, and those fragments would generally insert into the mouse genome randomly by a head-to-tail array. So this allowed us to add a gene, and very often we could do, it was useful for dominant mutations. We could create a number of disease models doing that. We could express different genes to monitor expression like GFP. So that technology was useful to that point. I should mention that again, the insertion was random, generally in a head-to-tail array, and the expression of the transgene often was determined not by how many copies were inserted, but by the genomic location that the insert, where the insert landed. To take a more targeted approach and actually be able to target a gene in the mouse genome, we needed to use mouse embryonic stem cells. So that's the targeted approach that really was happening a lot in the 1990s and into 2000s. And again, that required mouse embryonic stem cells, which added a lot of time to the process because the mutations were made on the stem cells. Those cells were then cultured and had to be injected into mouse blastocyst embryos to create chimeric mice, which then were bred to germline, to get germline transmission. But this allowed us to be able to target individual genes in the mouse genome, which was an advancement, to what we were doing before in terms of just gene addition. One of the challenges with doing gene targeting was that when you were working in mouse embryonic stem cells, you were restricted to strains for which there was a robust embryonic stem cell available, and that wasn't most of the strains. So a lot of the work was done on the 129 background, and then there were black 6 embryonic stem cells that were developed. But what would happen is that if you wanted to have a mutation on, for example, BALB-C, you would create it on black 6 and then do extensive backcrossing to get to the BALB-C background, which added an additional 1.5 years, even if you use speed congenics to get there. So people were looking for better technologies to do gene targeting, and the next generation of technologies included site-specific nucleases, of which CRISPR is one of them. And in addition, zinc fingers and TALENs are also site-specific nuclease-assisted genome editing. And the way these technologies work is that you create a double-strand break, which can either be resolved by non-homologous end joining, or in the context of a targeting vector, in the presence of a targeting vector, homology-directed repair. So non-homologous end joining allows for the deletion of 1 or 2 nucleotides, the insertion of 1 or 2 nucleotides that throws off the reading frame of the protein that you might be trying to modify. And again, in the presence of a targeting vector, you could also get homologous recombination. The way CRISPR works is on this slide, and if you look at the right side of the slide, the little cartoon, you can see that CRISPR works by, we create a single guide RNA for which 20 bases, I should say, are actually homologous to the sequence in the genome that we're trying to target. So that's 20 base pairs. We can, in the genome, but the trick here is that those 20 base pairs have to be next to the PAM sequence, or the photospacer adjacent motif. And that's 3 bases, and for the system that we're primarily using, strep pyogenes, those 3 bases are NGG. So now those 20 bases, you have to find 20 bases that are next to an NGG sequence, and then that single guide RNA in complex with the Cas9 protein will specifically cut 3 bases from that PAM sequence. And again, that double-stranded break can be resolved either by non-homologous end joining or by homology-directed repair. And the reason that we were so excited, that a lot of people are so excited about the CRISPR technology, is that this gives us, it's around 100,000-fold increase in efficiency when you're thinking about homologous recombination compared to the standard targeting in ES cells that we used to do so frequently. So because it's so efficient, it allows you to bypass embryonic stem cells, which shortens the timeline and also allows you to do this work with no selection elements required. So when we used to work in mouse embryonic stem cells, we were always using antibiotic resistance genes to screen for cells that actually got the targeting vector. In this case, we don't need to use those, and we don't need, therefore, to remove them, and it allows us to make precise single nucleotide changes without leaving any extra sequence. And when I say without leaving any extra sequence, in general, we used to surround our antibiotic resistance genes, NEO, for example, with LoxP sites or FRT sites that then we could remove the NEO resistance gene, but we'd still have that LoxP site there. We don't generally need to do that when we use CRISPR technology because it's so efficient. And again, we can go into single cell embryos instead of having to use mouse embryonic stem cells, which actually enables us to do this work in many more strains without having to do extensive backcrossing. So I mentioned that there were 3 different technologies that all cause nuclease-assisted double-strand breaks. And the reason that we favor CRISPR-Cas9 versus zinc finger or TALEN is that zinc finger and TALEN to target another gene in the genome, you have to engineer the molecule, the protein, the zinc finger protein or the TALEN protein, and then you have to test to make sure that it's actually targeting what you want. Whereas the specificity in the CRISPR system comes from the RNA, and it's very easy to synthesize another guide RNA. That takes a short period of time, and it really opens up this technology, makes it a lot easier to use. This technology gives us the potential to target multiple genes, which is really important when it comes to if the genes are tightly linked in the genome on the chromosome. And we can target, again, in single-cell embryos, and no need to use ES cells, which means that we can go into many more strains than we used to be able to go into directly with mouse embryonic stem cells. And again, small deletions we make through non-homologous end joining and targeting. We use oligo targeting vectors or larger plasmid targeting vectors for homologous recombination. In terms of timeline and costs compared to the conventional gene targeting in mouse embryonic stem cells, there's a big advantage with CRISPR. And I have on this slide the different types of projects that we perform using CRISPR technology. So indels are just small deletions or insertions, 1 or 2 bases added or deleted. When on this slide where it says deletion, that refers to using 2 guides, 2 different guide RNAs, and actually deleting the sequence between those 2 guides, and then using an oligo targeting vector, we can add a SNP, a single nucleotide change in the genome, or using plasmid targeting vectors, we can add in a reporter gene or create a conditional knockout. And the costs are quite a bit less for doing these compared with ES cells. 81% for an indel, that much less. And for the most difficult type of project that we do right now, the conditional knockout, it's around 38% less. And from a timeline perspective, for conventional gene targeting, it generally takes a year to a year and a half to get to germline transmission. I put 12 months as the baseline here. But you can see for these other types of projects, to get germline transmission is 6.5 months to 8 months, depending on the complexity of the project. So we've had a lot of experience doing these different types of projects. So small deletions, knockouts by indels, knockouts by deletion with 2 guides, point mutations, knock-ins of cDNAs or conditionals. And based on our experience, you can see that we are comfortable. The projects that now are highlighted in red are projects that we're comfortable performing for the external scientific community. And I should have mentioned this, but at JAX, we function as a core for JAX investigators. So we've been doing this work for, we did this work for several years prior to actually being able to offer this capability to scientists outside of JAX. And for scientists outside of JAX, we're very comfortable doing the projects that are highlighted in red. And if these projects, are standard, and if there are no concerns when we start the project about embryonic lethality for that particular gene, for example, then we will guarantee that if we accept the project, that we will that we will get you a mouse or you won't pay. And in addition, for some of the projects that are a little bit harder, they are feasible in mouse embryonic stem cells, and we have a process where we use CRISPR to do that at this point as well. In terms of our process and workflow, in general, the gene design, the guide synthesis, for indels, we go straight to microinjection, and then we have to characterize the animals. And we do that for indels. It's generally a sequence analysis to determine whether they are missing or have gained a nucleotide or 2. In terms of once we have founders identified, we breed them to get what we call the N1 generation. So these are now animals that have come through the germ line. And we can do that by speed expansion, and I'll mention a little bit more what speed expansion is. That could give you more N1 animals and move your project forward faster. We also validate that generation of mice is exactly what you want as well by sequence analysis prior to making an assay that is higher throughput for subsequent identification of the mice in the strain. For deletions with 2 guides, as well as any project that requires homologous recombination, our process is a little bit more extensive in that we add a guide evaluation step that we do in vitro in a cell line. Of course, we have to also create the targeting vector, so do some molecular biology there. But then it becomes microinjection and analysis of the founders. And the analysis for a homologous recombination experiment is generally more extensive. And when I get to the case studies, I hope to illustrate the different ways that we evaluate and validate that we have the mice that we think we have. Again, speed expansion is an option both at the founder breeding stage and for breeding the subsequent N1 animals. And just a quick word about speed expansion. What it is that we perform IVF, and generally we can start if we have a male founder or male N1 animal. It allows us to just take the sperm from 1 or 2 males and take females. Generally, we can do this with wild-type females from the appropriate background strain, and that allows us to get a large number of animals for the next generation. So that's something that we can do that actually can speed up the process, basically, so that it can take time off the long-term, the breeding that needs to be done to generate animals for subsequent experiments. Now, various inbred strains have been used for different, for performing different preclinical models. And I put this slide in this deck just to illustrate and to mention that at JAX we have access to a lot of these different strains, but also we have the capabilities of working on these different strains and going directly and making mutations directly on these backgrounds. And if you've been doing preclinical work in an NOD background, for example, because you're studying diabetes, it makes sense to be actually creating mutations on that same background as opposed to creating mutations and then maybe back-crossing it to that strain. And we have a lot of experience going into those different strains, and we also have access to, at JAX, the various strains. We have a very robust genetic stability program, which allows us to really have well-annotated genomes so that that genomic information is important when it comes to designing the guides and the targeting vectors that we need so that we have a really efficient homologous recombination and double-strand break process. The other thing I would mention is that we have extensive experience with these strains in terms of their reproductive biology. So we've used them a lot. We know how many embryos we're likely to get, and we scale the experiment appropriately based on that. These are just a short list of a lot of the strains that we've worked on, but we've successfully modified 74 different mouse strains to date, which is, in my mind, a number. I doubt that there are very many groups who have approached that kind of number. So it's been a privilege for me to work at JAX and to work with people who have this experience. It's been really great. In terms of our experience making different types of models using CRISPR technology, are going towards more sophisticated models that require plasmid targeting vectors. But we still do get deletion projects, requests to do those, as well as some Indel projects. But we're seeing that most of the work is going more towards oligo and plasmid targeting vector work. And our success rate has been really good. It's been 97%, and that's for the external projects that we've done in the last 2 years since we launched this externally. I'd like to switch gears a little bit and talk to you about a couple of case studies that we've done. Case studies for projects that we've performed. And this will allow you to see that what the goals were and what strategies we came up to create those mice and to also validate them. And I want to mention that these case studies were done for internal faculty, and we have permission to talk about them. And every project that we do is proprietary for the group that we perform it for, so we can't talk about the work that we do without permission. But these projects that I'm going to tell you about we have permission to talk about, and they were performed for one of the faculty members at JAX. And the first one is we created an allelic series for familial ALS, and we used an oligo-mediated knock-in strategy to do that. So it was known that there was an abundance of non-synonymous SNPs in humans in human exon 4. And based on that, we set out to create an allelic series. So 4 different SNPs we wanted to make in a model for 4 different projects, basically. And the one that I'm going to focus on is the second one from the left on this slide. It's an arginine to cysteine change. And these different changes, but the strategies that we used for this are similar for all of the different 4 projects, but I'm really only going to talk about one of them. So the equivalent human codons reside in mouse exon 4, and we set out to create those mutations, again, using a single-stranded DNA oligo strategy, which is good for introducing SNP variants, for rapid generation of the template. Genotyping the founders is not as complex as when you use a plasmid targeting vector, and I'll explain that a little bit more moving forward, but generally we're doing a lot of sequencing for these types of projects. The challenges can be whether we can find good guides in the region that we're trying to create the mutation in. And again, when I say whether we can find good guides, we want to find 20 base pairs, 20 bases that are next to an NGG sequence to create the guide. We often, you'll see, try to create a silent mutation to reduce the recutting, and I'll try to explain that as we move forward. So this slide shows 3 possible guides that we were evaluating to create this arginine to cysteine mutation. And to do that, we were actually mutating a C to a T. So the CGT goes to a TGT. And this was the first guide, second guide, and the third guide. And what I'm going to point out is that when I go back to this second guide, that the introduction of that T mutation, that C to T change, actually destroys the PAM sequence, which is in purple as opposed to blue, those 3 bases. And because that sequence is disruptive and changed, the guide Cas9 complex can't come back and recut once we have the mutation that we want. So that's something that we try to do, and that's the guide that we used for this project. So on this slide you can see that we create a single-stranded DNA template that has the 1 base change that we want, and it has around 60 base pairs on either side of it that are homology arms, and that the correct mutation destroys the PAM sequence and prevents additional Cas9 DNA cleavage. Before I move forward and show you data from this project, I want to talk a little bit about mosaicism, something that we see when we use this technology. And just to show you what that means, on this slide you can see that when we inject the reagents, and by reagents I mean the targeting vector, the guide, and the mRNA that expresses Cas9, we can get the activity from that mix happening at the 1-cell stage, which is what we hope for. But also often those reagents hang around, and we can get activity at 2-cell and at 4-cell. And what that means is that we can get multiple alleles from one founder mouse, not just 2. So what does that mean? In a picture form, I'll go to the next slide, which shows you an example of this. In this example, they were trying, and this is not something that we did that was done at JAX, but Yen did it and published it in Developmental Biology in 2014. They tried to mutate the tyrosinase gene, which is required for pigmentation. And these are all potential founders, and you can see that there are some mice that are completely white, which means that the mutation that they were trying to make happened at the single-cell stage, so you can see it in almost all the cells. But there are some mice that are mostly agouti here, but have a little bit of white, and there are some mice that have a little bit more white, but still have significant amounts of agouti, and this just illustrates that these events, these genetic events, happen not at the 1-cell stage, but at 2 or 4 or somewhere after that. So now to get back to the project, what we saw in potential founder 56-4, and this is a Sanger sequencing trace that you can see on the bottom, and you can see that it's clean in the sense that there are single peaks, except for, if you see the arrow that says C to T, there's 2 peaks there. There's a small peak that represents that C to T change, and a bigger peak that just represents the C. And so the way we interpret this is that this is a low-frequency T allele, so it's a mosaic animal, but we do have some cells that have what we want, but it's clean. There's no other mutation there. Now, the next potential founder, 56-1, you can see that the sequence is clean up until that C to T change, but then after that, you can see that there's multiple peaks in the same areas, indicating that there's probably an indel in that mouse as well. So we have a high frequency T allele, but there is the desired mutation plus indels, and what we don't know is whether that indel is on the different chromosome from the mutation that we want, or whether it's actually insists on the same chromosome. So what we do is that we topoclone the PCR mix and sequence the individual clones to determine whether we have the T and the mutation that we want and the indel on different chromosomes, or whether they're on the same chromosome. If they are on the same chromosome, we generally won't breed that mouse because it's not going to be able to segregate the two mutations away. If they're on different chromosomes, we go ahead and generally have been successful at getting the desired mutation. So to summarize what we've done on this project, we were able to rapidly produce founder animals carrying these single nucleotide variants. And we were able to do it without leaving any residual DNA modifications like LoxP sites or FRT sites, which we would have had to leave there had we been doing work in mouse embryonic stem cells. And these mice are now being evaluated phenotypically by the investigator at JAX. So that's the first case study. And at this point, I'd like to move on to the second case study, which was a model for Friedreich's ataxia, and we created a conditional allele for taxin using a double-stranded DNA plasmid-mediated knock-in strategy. A little bit about Friedreich's ataxia. It's a progressive degenerative disease causing neurodegeneration and cardiomyopathy. And the taxin gene encodes a mitochondrial protein involved in iron transport and respiration. But what was known before we started the project was that this gene had already been knocked out, and animals that were homozygous for this null mutant, it was associated with embryonic lethality. So we had to create a conditional mutation and use the double-stranded plasmid strategy, knock-in strategy to do that. And this is good for introducing large reporter genes or creating conditional alleles. It's more cumbersome to create these plasmids than to just synthesize a single-stranded oligo, so that's one of the challenges. In general, the homologous recombination frequency is lower than with a single-stranded oligo strategy, and the genotyping is a lot more complex than what we have to do to characterize indels and just straight deletions or SNPs. So our goal was to flank exon 2 with LoxP sites. And to do that, we try to find sequence that low sequence conservation in the introns, because we don't want to affect anything that could affect the levels of expression, and we hope to be able to identify guide RNAs again in the selected region, which we were able to do in this case. So the targeting vector is going to look like this, where the thin blue lines represent the LoxP sites. The arrow is the exon 2. And so you can see that we have LoxP sites around exon 2, and then we have around 1.5 kilobases of homology on either side of those LoxP sites. So if you look at the wild type exon 2, we were able to identify possible guides upstream and downstream in the introns surrounding exon 2. And what we did, the way we designed this was that we put in, if you look at the floxed exon 2, which is towards the bottom of the slide, you can see that that dark blue sequence, which represents the LoxP sites, if inserted properly, cannot be cut by those guides anymore because it basically breaks up the sequence of the guide, and that's both the guide upstream and downstream of exon 2. So, and again, we had one guide that we found upstream and two possible guides that we found downstream, but they were both, by the strategy that we used, disrupted by the insertion of the LoxP sites so that there couldn't be additional cutting. I wanted to talk about our genotyping strategy for projects that involve plasmid knock-ins. And in general, what we do is a short-range PCR, and in this case just to determ' whether the LoxP sites are in the genome. And then we do long-range PCR on both the 5' and 3' end, or the left and the right homology arm sides, and where one primer is outside of the arm and another primer is within the targeting vector, but it includes the LoxP sites as well. And what this means is that we can't get a long-range PCR product that has LoxP sites unless those LoxP sites are at the right, are inserted correctly in the genome. So the other thing that we do, so in other words, the first short-range PCR just tells us that these are mice that we should screen further. The long-range PCR shows us whether it's targeted properly. And the other thing we do is we do an assay for the vector backbone to determine whether the targeting vector randomly inserted, because if it randomly inserted, it's likely that that vector backbone would still be in the genome. So we've done that. We do that routinely. And I should mention that this strategy, we've done it as well as done Southern analysis and gotten consistent results using both of those technologies. I just want to show you some data for some of these potential founders that we got from this project. And I'm going to focus on founder 46.2 and 46.5. And what we're looking for here in the short-range PCR is we expect to see two bands, one that is the size of wild-type DNA for that locus, and then one that's a little bit bigger that accounts for the size of the LoxP sites that we've inserted. And you can see that in 46.2 there are two bands of this. One band that's a little bit bigger than the smaller one, and I'll show you some sequence data for that in a couple of slides. And for 46.5, and let me just click through this, you can see that there's only one band, and it appears to be actually higher than our wild-type band. So let me go to the sequence data, and I should have mentioned on the previous slide that those PCRs that we do are a major part of our strategy, but we also, to validate that the mouse is right, are doing extensive sequencing analysis to make sure that the change that we're trying to make is actually there and it's what we think it is. So this is a Sanger sequencing trace for founder 46.2, and you can see that when we deconvolute the sequence, we have what looks like the wild-type sequence as well as the LoxP sequence, which would indicate that one of the chromosomes had the homologous recombination event and the other one did not, remain wild-type. So this is a founder that we can breed and get the desired mutation with the LoxP sites from that. Now for 46.5, where we saw that one band that looked like it was larger, we have a very clean Sanger sequencing trace, and that trace is consistent with the sequence being that of the LoxP site, not of wild type. So it actually appears, and this is a rare thing that happens, that we were able to get homologous recombination in this particular founder on both chromosomes. And again, that doesn't happen that frequently, but in this case, it did. So to sum up, we were able to rapidly produce founders carrying the LoxP insertions. They were properly inserted. It's important, I can't stress enough, that thorough upfront molecular genotyping in founders is really important, as well as doing the same kind of analysis on N1 animals to make sure that you're using, that there really is nothing, that you really understand what's going on with the animals that you're going to be using for subsequent preclinical testing. And these animals are currently being tested with tissue-specific CRISPR strains. Wanted to talk a little bit about about CRISPR and off-targets. When we design our strategies, each guide is given an off-target score. We try to use guides with the lowest off-target score. In the event that we can't find guides with low off-target scores, we recommend an off-target analysis in the N1 mice. And I should mention that what I'm talking about here are predicted off-targets. In general, we evaluate where these other off-targets could be, and in general, they're not closely linked with the mutation that we're trying to make. We just need to know that they're there so that we can make sure that we're segregating them as we breed the animals. So we don't worry that much about off-targets of this sort. We have analyzed from 7 different projects that involve 6 different genes, and when we look at the different off-target scores, the bottom line is that when the off-target score is below 2, or when there's a non-canonical PAM sequence, we don't see any off-target events. However, when you have a canonical PAM and a higher score than 2, we have been able to see events. I will mention that I know that there has been a publication probably around a month ago in Nature Methods that indicated that they found single nucleotide variants and a large number of them in a CRISPR experiment that we did. And we are currently doing some work to evaluate whether we can reproduce that same result in some of the animals that we've already made. But we do have questions about that paper in terms of the lineage of the different of the mice that they analyzed versus the control. And we note that there is no known CRISPR mechanism by which single nucleotide variants would come up. So we do have a lot of questions about that, but we are following up, and I know a number of groups, in addition to JAX, are also following up on that publication. I wanted to talk about some of the things that we've learned by doing the number of projects that we've been able to do. And we do a large number of projects, not only for our faculty, but for people outside of JAX. One thing that we found that we didn't anticipate originally is that we've often been asked to create single nucleotide polymorphic mutations in genes that may be associated with embryonic lethality. And even though we're making heterozygotes, what we find is that sometimes during the process, we can create the creation of indels, knockouts, can actually make it more difficult to get the desired mutation that we're trying to make. So we've had some experience trying to titrate the reagents to try to favor the homologous recombination of our oligo insert versus the indel formation. And that's been something that we didn't really anticipate when we really started doing a lot of these projects. Another challenge comes up often, not that often actually, is low efficiency cutting, and fortunately that doesn't really happen that often. Strain-specific reproductive biology limitations just refers to the fact that different strains behave differently in terms of their ability to withstand an injection, and also in terms of the number of embryos and the quality of embryos that you're able to generate from them. Our experience in reproductive biology is really helpful in helping us scale the experiments appropriately when we're working on various different strains. Potential impact of DNA variation on gene editing experiments. Just as you might expect, it's important to make sure that you are designing your experiment consistent with the genome that you're trying to target. So just because you have a guide that might work well in Black 6, there might be a polymorphism in another strain, so you should be aware of that and create the appropriate guide for the appropriate strain. And one last thing that I wanted to mention is that we've seen random insertion of the targeting vector when we've done experiments involving plasmid targeting vectors. In general, this hasn't been a big problem because we've been able to, upon breeding the animals, those plasmid that have been inserted improperly in the wrong place have been segregated away. So hasn't been a very big issue, but it's just something that if you're doing this in your own lab, you want to make sure that you're aware of. So I hope I've been able to communicate to you that CRISPR can generate new mouse models for faster and cheaper than ever before. That the success really depends on really thorough characterization of both the founder and N1 animals. And at JAX, we really bring, I think, the genetic engineering, the reproductive science experience, the animal husbandry expertise, to model generation, as well as the access to almost 9,000 different mouse strains. And this technology really opens it up to allow you to work on a lot of different mouse strains. I want to thank my colleagues at the Genetic Engineering Technologies Group led by Bill Buis. And I'd also like to thank Kat Lutz, who's the investigator at JAX, who allowed us to talk about both of those projects that we performed for her group. Thanks, and I'll take any questions that you have.
Thank you, Dr. Grass, for your presentation. A quick reminder for our audience on how to submit questions. Simply type them in the Q&A box found by clicking on the green Q&A button at the lower left of the presentation window. Dr. Grass will answer as many questions as time permits. And the first question is, do you have a lot of experience creating conditional mutations?
Yes, we actually have. We get asked to create conditional mutations a lot. And I would say, being at JAX, one of the advantages that we have is that we actually have access to a lot of the different CRISPR strains that you might want to use subsequent to having your conditional reel. And I will remind people that it's important to match the background strain. And so having access to all those CRISPR strains, making sure that if you're creating the model on a Black 6J, that your CRISPR strain is on Black 6J, that's important. And in general, as in the case study that I showed, I think the second one, we generally use the strategy of using a large plasmid targeting vector to create those mutations.
Are you concerned about off-target events, and how does JAX address this issue?
We try our best to design around all the predicted off-targets that you could have. So when I say predictive off-targets, each guide that we have is given a score and we look at the different potential off sites where they be creating double-stranded cutting in the genome. And if we feel that it's likely that we might get an off-target event, we will suggest that we do off-target analysis to determine whether that happened or not. But in general, those events can be segregated away. And then I think I mentioned this during the presentation, but there has been a report that there may be other mutations associated with the CRISPR technology. Some questions about that paper in terms of the lineage of the mice, the mechanism by which they think that this happened, because I don't think there is a known mechanism for how CRISPR would facilitate single nucleotide changes. And I will also mention that those nucleotide changes weren't predictable, meaning they weren't associated with any particular sequence. So we think that there's a chance that this may be explained by just genetic drift between the founder animals that were evaluated and the reference genome and control mouse that they were used. But at any rate, we are evaluating whether we're seeing that same thing in our mice, and that evaluation, hopefully we'll have some data in the next couple of months on that.
Looks like we have time for one more question, and it is, do you think CRISPR will eliminate the utility of randomly integrated transgenic lines?
I would say that I think that there's a place in the toolbox for all of these different technologies. And I think that randomly inserted lines have been used and effectively to study various things. Again, for dominant mutations, to look at the expression from particular promoters and use a gene like GFP to tell you where expression is happening. I think it's been valuable, and also to express genes in a deregulated manner. I think that if you're looking to get a variety of different expression levels in mice, then actually that transgenic approach, that random approach, may be useful, because you can create those mice, evaluate different founder lines, and each one of them may have different expression levels, and then you can choose the ones that best suit your research. So I don't think it'll eliminate that, but I do think that it's a great tool that really has allowed us to move forward and create a lot of different new mutations quickly. Thanks.
Well, I would like to once again thank Dr. Grass for his presentation. Do you have any final comments?
No, I would just like to say it was a pleasure to be able to be here. I thought that I hope that this presentation, it was informative and helped people in the audience who are either doing it in your own lab, or if there's any way that we at the Jackson Lab can assist you, we'd love to be able to discuss that with you.
Well, I would like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today will be addressed via email. We would like to thank our sponsor, the Jackson Laboratory, for underwriting today's educational webcast. This webinar has been approved for continuing educational credits. Please click on the CE button at the bottom left corner and follow the process to receive your credits. This webcast can be viewed on demand through January 21st, 2018. LabRoots will alert you via email when it's available for replay, and we encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.